Debunking land animal to whale evolution. Atheists claim that a semi-aquatic land-based creature evolved into whales. Allegedly, a wolf-like, or dog-like or hippo-like or bear-like creature evolved into whales by random chance and natural selection. As evidence for this atheist creation myth, they point to fossils of alleged intermediate creatures such as Pakicetus, Ambulocetus, and Basilosaurus. In this video, we will debunk this atheist claim. We will show that 1. The logic of the atheist claim is false, and 2. Taxonomically restricted genes falsify land animal to whale evolution by random variations and natural selection. Item 1. The atheist logic is false. 1. Similarity does not prove common descent. Evidence. Atheists believe that convergent evolution can create similarity that is not due to common descent. 2. Fossils do not appear in the record with birth and death certificates, so there is no way to determine parent-child relationships between fossils. 3. Fossils cannot establish that changes arose by random chance and natural selection. 4. Without a functional mechanism, an appeal to evolution is an appeal to magic. The evolutionary literature is full of such appeals to magic, where allegedly scientific papers claim that something was created by evolution without providing any biochemical step-by-step mechanism to establish that claim, that something arose by random chance and natural selection. 5. So, to claim that fossil creature B arose from fossil creature D is nonsense. 6. And to claim that fossil creature B arose from fossil creature D by random variations and natural selection is nonsense. 7. So, morphological intermediacy does not establish evolutionary intermediacy by random chance and natural selection. Conclusion 1. Therefore, the atheist logic is false. Item 2. Whale features. Whales possess a variety of unique features required for life in water. These include blowholes on top of their heads, large horizontal tail flukes that move up and down for propulsion through water, fully aquatic locomotion with streamlined bodies, and flippers for steering, a thick layer of blubber for insulation, energy storage and buoyancy control, echolocation in toothed whales, specialized ear structures for hearing underwater, the ability to hold their breath for long periods of time, e.g., an hour for some species, due to highly efficient oxygen storage and utilization systems, a diving reflex that conserves oxygen and reduces heart rate during long dives, fully aquatic reproduction, giving birth and nursing in water. Calves are born tail first to reduce risk of drowning. Whales possess genes and genetic logic circuits required for each of these unique traits. This leads us to taxonomically restricted genes in whales. Item 2. Continued. Taxonomically restricted genes falsify land animal to whale evolution by random variations and natural selection. Comparative genome studies show that every taxonomic group has 10 to 20% of genes that are taxonomically restricted genes, TRGs. These TRGs are required for morphological specifications that are unique to that taxonomic group. Taxonomically restricted genes are genes that are unique to a particular taxonomic group and have no recognizable homologs in other groups. Comparative genomics studies in whales and land-based animals, such as hippos, genomic comparisons have revealed thousands of genes that are unique to either class, contributing to their respective physiological and developmental differences. For instance, whales possess unique taxonomically restricted genes involved in their unique traits mentioned earlier above. Whales have about 20,000 genes. Comparative genome studies show that every taxonomic group has 10 to 20% of genes that are taxonomically restricted genes. So, whales have 2,000 genes that are not present in land-based animals. Protein lengths are 300 to 500 amino acids long in mammals. Between 35% and 95% of amino acid sequences are conserved in mammal proteins, depending on the protein. This means that a lower bound of about 100 amino acids are conserved, needed for function, in mammal proteins. Protein background. Empirical research shows that 1 in 10 to the power of 63 amino acid sequences forms a functional 92 amino acid protein structure. We will use this number for our 100 amino acid calculation mentioned above. Reminder. 10 to the power of 63 means 10 to the power of 63, i.e., 1 followed by 63 zeros. This can also be represented as 1e63. Protein functionality has to arise by random chance mutations, e.g., in de novo gene origination, natural selection can only act on what is functional. So, to explore amino acid sequence space, we need 10 to the power of 63 attempts to find one functioning 92 amino acid long protein. The mass of Earth's entire biosphere is 2 times 10 to the power of 15 kilogram. Amino acids are about 75 to 200 atomic mass units. So, if the entire biosphere is composed of amino acids, that would provide us 1.6 times 10 to the power of 40 amino acids. The age of the Earth is about 4.54 billion years, so 1.43 times 10 to the power of 17 seconds. The fastest bond-breaking or formation reactions take about 10 femtoseconds. We can use this information to calculate the total number of trials available in the biosphere over the history of the Earth. That number is 1.6 times 10 to the 40 times 1.43 times 10 to the 17 times 1e14 equals 2.3 times 10 to the 71. We will round this up to 3e71. For random chance to find one functioning protein, of a length matching the conserved lengths we observe, would take 1e63 trials. To find two independent proteins would take 1e126 trials. To find 3 independent proteins would take 1e189 trials. However, the number of trials available in the entire history of life on Earth is only 3e71. This means that only one new functional protein, of length 92 amino acids or higher, can be found by random variations in the entire history of life on Earth. And if random chance does not find the protein, natural selection cannot act on it. Application: to evolve whales from land-based semi-aquatic creatures, like hippos or wolf-like creatures, we need to generate 2,000 new taxonomically restricted genes, and all of the new genetic coding circuits and logic needed to use those 2,000 new genes. However, the entire biosphere of Earth has the time and material resources to create only one new functional TRG protein. This is far short of the 2,000 new genes that are needed to evolve whales from land-based semi-aquatic animals. Therefore, taxonomically restricted genes falsify land animal to whale evolution by random variations and natural selection. Conclusion 2. Taxonomically restricted genes falsify land animal to whale evolution by random variations and natural selection. Thanks to Hans Weichselbaum for requesting discussion of this topic. Thanks for watching. Please like, share and subscribe, and comment below if you agree or would like to add your thoughts.